of course, which may arise due to change in the base sequence of the gene you call gene mutation, may also result in the higher organisms, the change in chromosomal numbers or their structures. You call it as chromosomal mutation. And of course, the history, people who have contributed in this particular field, who worked at goes back to 1791, where an English farmer he found that a male lamb with usually short legs. Of course, he was not aware what is the thing. So the term mutation was introduced by Hugo de Vries in the 1900s. He was the first person to use the term mutation. Then um, of course, different groups started working on mutation. Uh, One group was working in Drosophila. It was Morgan's group, who were working on that. Uh, then Muller, he, of course, showed the mutagenic action of X-ray in the year 1927. Of course, there also he used his experimental thing was Drosophila. Of course, Miller was awarded Nobel Prize in Physiology in 1946. In 1929, Stadler described the mutagenic effect of X-rays in garlic. So the characteristics of mutation. Of course, mutations are random events in terms of time and occurrence and the gene in which they occur. Most are harmful, but some are beneficial. Mutations may be recurrent, so the spontaneous mutations occur at low frequencies in nature. So this point must bear actually the evolution of the organism, starting from the thing primitive cell. From that, when present day organisms are evolved, that particular thing is due to introduction of this change. Of course, 1 in million it happens. So this is the things. Rate of spontaneous mutation varies from one to other gene. Some genes show high rates of spontaneous mutations are known as mutable genes. Some others suppress the mutation rate of other gene called anti-mutator genes. So mutation at one particular thing, it suppresses the mutation in the other place. So mutation producing same phenotype effect may be located at different sites within the same gene. Some sites within the gene show high rates of mutation at sites called hotspots. Actually, so the hotspot, the where so like thing, like the frequency of mutation, it is very high. So they use the word hotspots. It can occur in any tissue or cell of an organism, and mutational events can occur in any developmental stage of the organism. Of course, it need not confine to the things, in any stage of an organism it may occur. Mutation occurs in both forward or reverse directions. So the things, in the double-stranded DNA, if like the strand which is in 5 prime 3 prime direction, and the one in the reverse direction, in both the things it may happen. Rate of forward mutations are higher than those of reverse mutations. You must remember this. So they are quite often found in forward directions. But most mutant alleles are pleiotropic. So then the mutagens, there are of course basically two kinds of physical agents and chemical agents. The physical agents, UV rays, X-rays, gamma rays. Of course, among them, these gamma rays are so. Of course, like highly, like um, they're risky. Uh, you know, they cause mutation. Um, but these gamma rays are useful for in medical, in you know, in medical treatments. Of course, the radiation, it is used there also. Of course, only thing, cancer treatments it is used. Of course, one side, you have to get rid of the cancer cells. At the same time, ಏನ್ ಮಾಡ್ತಾರಪ್ಪ ಇವರು. So knowingly, no, you are using gamma rays. Of course, chemical mutagen, a large number of chemicals, like of course, yesterday we were discussing this. Most of the recently course, not recently means 60s onwards. The agricultural chemicals, insecticides, fungicides, herbicides, so it were made available, things. Many of them are known to cause mutation. So I told you actually this is the reason, especially coastal and Malnad region, the cause for increased cancer thing, like disease. Mainly it is due to insecticides, fungicides and herbicides that were so introduced. And of course people, they are also not bothered about the things, like how to use, so what quantity they have to use and all that. So the like excess also many people they try to use. That's the thing. Then the next thing is the other chemicals which are used in molecular biology studies: ethidium bromide, acryflavin, acridin, orange, and etc. They are also actually mutagenic, like this ethidium bromide, which is used in gel electrophoresis DNA. Actually, people who are doing have to be very careful. So that particular thing. Um,
+2 तक के
uh, Causes of mutations: errors in DNA replication, second, errors in DNA recombination, both things, so the replication, simple things, the recombination, which is part of the sexual life cycle. In both the things, like so the mutation causes errors in DNA replication as well as DNA recombination. Then of course, chemical damage to DNA. The DNA, many chemical mutagens, some exogenous, some man-made, some environmental, are capable of damaging DNA. Many chemotherapeutic drugs and intercalating agents, drugs, they function by damaging DNA. Then radiation, gamma rays, X-rays, even UV light can interact with compounds in the cell, generating free radicals which cause chemical damage to the DNA. Free radicals. And the types of mutations: the mutation, the different types based on the cytological things, the gene mutation and chromosomal mutation, they are broadly classified. Then based on effect of phenotype or tissue of origin, they are classified as somatic and genetic mutation. Genetic mutation is also called as germline. Somatic mutation and the genetic mutation or germline mutation. Then based on the cause of their origin, so they are called as spontaneous mutation and induced mutation. Induced mutation. Then types of traits, based on the types of traits, so the visible and biochemical mutations. So like that, they are classified into different things. The first one, based on effect of tissue of origin, somatic mutation. The first one, arise in the somatic cells, passed on to the cells through the process of mitosis. Effect of these mutations depends on the type of the cells in which they occur and the developmental stage of the organism. It occurs early in the development. If it occurs early in the development, larger the clone of the mutated cells. Of course, the points here, the somatic mutation, of course, it happens, um, like in the cells, through mitotic division, it happens. Um, and here, the cell to cell, it may be transferred. During the growth of the organism, when there is cell multiplication, it may happen. But suppose if that particular thing, part where that change has occurred, and in case if it is thing, a crucial things, then the effect would be quite high. The sensitive organ, if it is like mm, and also stage of the organism also it matters. Then the genetic mutation or germline mutation occur in the cells that produce gametes passed on to future generations. The multicellular organisms, the term mutation is generally used for germline mutations. Um, the term mutation is generally used for germline mutation in the higher organisms. And based on the cytological basis, you classify mutation, things into chromosomal mutation. First type, there are four types of chromosomal mutation may occur. Deletion, a part of the chromosomal things may be deleted, or inversion. It may then translocation, duplication. In the next pictures, we will try to see. When a part of the chromosome is left out, it is deletion. When it is removed, as the arrows show, thus that part is lost in the things. Thus, the function, it affects the functions of these chromosomes. So like that particular things, it is lost, the genetic information in that particular region. Second thing is, when part of a chromatid breaks off and attaches to its sister chromatid, an insertion occurs that results in duplication of genes on the same chromosome. So the insertion, so that means to say duplication. The information of a similar kind is increased many times. So that is duplicate, insertion. It happens due to insertion. Then another thing is when part of a chromosome breaks off and reattaches backwards, an inversion occurs. So here, what happens? The thing, like part of the chromosome breaks off and attaches, in backward direction. That is inversion, So the information, there is no continuity of the information occurs here. The information, continuity of the information is lost here. That means to say the function of these chromosomes, it is impaired. Then when part of the chromosome breaks off and is added to a different chromosome, a translocation occurs. Translocation. So that means to say when part of one chromosome breaks off and is added to a different chromosome, a translocation occurs. So that means to say a part of the, like chromosome, one chromosome, it breaks off and added to another chromosome, that may occur. So these are all different ways, chromosomal mutation occurs. Then of course, how it happens and all, then the deletion have shown how it happens. Then inversion, how it happens, they have shown. Then duplication, how it happens, it is shown here. Then translocation, how it happens. Okay, that was chromosomal mutation. Next is gene mutation. Gene mutation.
Sir, what are transposon genes?
Huh?
Transposon genes, sir.
Transposons.
Genes, yes, sir.
ಅದು ಆಕ್ಚುಲಿ ಬಯೋಲಾಜಿಕಲ್ ಇನ್ನೊಂದು ಇದರಲ್ಲಿ ಅದು ಬರುತ್ತೆ. ಈಗ ಉದಾಹರಣೆಗೆ
English, sir. Sir, no Kannada. Sir, no Kannada, sir, in English, sir.
ಹೇಳಿ
Sir, in English, sir.
Yeah, tell me.
Sir, what are jumping genes, sir? Transposons.
We have covered, that the insertion sequences. Already it is dealt, in the two classes before. So actually they are called as insertion sequences. So which are, of course, while discussing, uh, like your conjugation, F plus type and HFR type. So HFR actually the thing, like the thing, like which is integrated with the main chromosome, the sex factor. Actually it is caused by the transposons, so the transposal elements. Then that means to say, it becomes thin. Of course, transposable elements, of course we have type. There are simple transposons and composite transposons. Simple transposons are, the one, which carry only the things, of course, how these particular things, are able to, jump their places, means, like change their places immediately, anywhere in the things, they can change their location, and they get themselves integrated. That is mainly because of the things, they carry information for that particular, transposase. So they have only that particular, simple transposons. Then there are other things called composite transposons. In addition to this activity, means ability to translocate anywhere in the things. So they do carry certain informations, like drug resistance and other things, informations they carry. So of course that which have been utilized. And of course just now, like we were talking about physical and chemical mutagens, the biological mutations, these transposons are mainly cause for many, of course, health problems. So like they're also, like involved in many these like mutations, mutation things, and of course they can be considered biological things. And in organisms like, you have Helicobacter pylori. Then you have, that is a bacterium, which causes gastric cancer. Yeah, gastric ulcer, of course, which later, like mm, like changes that particular thing into gastric cancer. Similarly, like you have certain viruses like hepatitis B, which causes the thing, like liver cancer, like that. Of course, so these are all some of the things, there. So you also have, these biological tools, mutagens, biological. You can add, so the physical mutagens, the chemical mutagens, and the biological mutagens, and in the that class, of course, your transposons can have a place. So anything?
Yes, sir. Got it.
Okay. Okay. Then in next thing is gene mutations, change in the uh, nucleotide sequence of a gene, and it may only involve a single nucleotide. So the types of these mutations are point mutations, missense mutations, nonsense mutations, and frame shift mutations. So when mutation occurs, a change in the nucleotide sequence occurs, so how it may be, like visualized, or manifestation, it may, like different ways: point mutation, missense, nonsense, or frame shift. The first one, point mutation, change of a single nucleotide, include includes the deletion, insertion, or substitution of one nucleotide in a gene. One nucleotide in a gene. Here, what happens? For this example, point mutation, sickle cell anemia. Sickle. That is the disease, rare thing. Of course, it is also known to be carried through uh, genetics. Genetically also it is known to be carried. And here, what happens? The change of a single nucleotide, you know, so that results in deletion, insertion, or substitution of one nucleotide in a gene. As a result, what happens? The thing, like the red blood corpuscles, instead of they are rolling out as a circular molecules, so they come out like this, in the picture. So the sickle shaped things, like canine shaped, sickle shaped. So that means to say their function is reduced. The people who are affected by this particular problem, they are actually, so they don't get, oxygen so much. So that's the problem. More oftenly, they have to undergo transfusion. Transfusion. Of course, they think, they can survive by continuously getting blood from the other sources. So they somehow they have to manage. So, but here, that's an example. Point mutation, where so the sickle cell anemia. So the things, like hemoglobin gene, a small change in the hemoglobin gene, it results in rolling out sickle shaped the red blood corpuscles. This sickle cell gene, a base substitution mutation, so that is shown here. The normal RBCs, so they are rolled out, whereas in the other case, sickle, the where the mutation has occurred in the like occurred in hemoglobin gene, then what happens? Like the it results in the formation of like half moon shaped or sickle shaped things, so which lack the of course they're not fully efficient, because they're subdued their activity. So like, so that's the thing, people having this problem, in their blood, sickle cell I know, like these red blood corpuscles. Then they will, so because they will not get oxygen thing, the sufficient oxygen supply through the in the blood circulation. So they have to opt for other means. Next, the missense mutation. This cause of the replacement of a single amino acid of polypeptide chain. So the cause of the replacement of a single amino acid of polypeptide chain means because triplet. Here the change occurs in a triplet. As a result, the things replacement of single amino acid in a polypeptide chain occurs. That means to say the polypeptide chain, the functional polypeptide chain, which is supposed to have a set of these amino acid molecules in the middle, an incorrect amino acid. For example, here they have shown no histidine in the polypeptide chain. If it is replaced by incorrect amino acid, that is proline, so which may produce malfunctioning of the protein. So that means to say, so the the protein function is thing it results in malfunctioning of the protein molecule. That is missense mutation. Next, nonsense mutation. Nonsense, it produces a nonsense codon. Such a codon does not code for any amino acid. Nonsense codon terminate the growth of a polypeptide chain. So, for example, here they have shown. So the things triplets like UAG, UAA, UGA. If any of these things like if they are introduced in the middle of the thing, the mRNA, during translation, that is the protein synthesis. If the mRNA carries the this information, so the nonsense mutation, of course, carrying any of these triplets, UAG or UAA or UGA. So suppose in the middle of the things, if it is there, then suddenly, during the translation, amino acids, sorry, protein synthesis, it results in thing, termination of the protein synthesis. So it is not complete protein synthesis. The fully functional protein is not thing, resulted. So that's the nonsense mutation. Next is frame shift mutation. Inserting or deleting one or more nucleotides. So changes the reading frame, of course, either deletion or insertion of one or more nucleotides um, happen, results in reading frame, change in reading frame. So like changing a sentence. For example, proteins built incorrectly. As a result, what happens? The frame shift mutation. So what happens is the the thing, because the information either by insertion or deletion of either one or more nucleotides, if they are thing, either removed or added, what the reading frame during protein synthesis, it is altered. So that is the thing. Then molecular basis of mutation. Mutation in any character is generally due to a change in amino acid sequence of the protein, determining the expression of that right? This change in amino acid sequence of the protein would obviously due to a change in the base sequence of the DNA molecule coding for proteins, which results in base substitution, base addition or deletion, transposition, or trinucleotide repeat expansion. So the first one, how it happens, base substitution. It is one of the it is one base in a DNA molecule. If it is replaced by another molecule, that is substitution. So one base in a DNA molecule, if it is replaced by another, then so it results in either transition or transversion. There are two types of things that may the results that may occur. So for example, base substitution is of two types. Transition, purine is replaced with a purine. Pyrimidine is replaced with a pyrimidine. It affects one codon and replaces one amino acid in concerned protein. Next, transversion. A purine is replaced by pyrimidine. A purine is replaced by a pyrimidine. So there in the previous case, purine for purine and pyrimidine for, but here what happens? Transversion. Purine is replaced by pyrimidine, or pyrimidine is replaced by purine. So as a result, what happens? The cause is transversion. The change in the the effect. One transversion affects one codon and replaces one amino acid in concerned protein. A base substitution usually leads to base pair substitution. Here, of course, by chance, base substitution, a single, base, if it is added, then what happens in the thing, first cycle of DNA replication, of course, it gets amplified, the presence of this, which is initially in the original thing, it is at one place. When they, during DNA replication, it happens, it goes on in a cyclic manner. Its thing, gets, increased. The effect will be more further. Then base addition or deletion. Insertion of one or more bases in a DNA molecule is called base addition. So loss of one or more bases known as base deletion. So these two leads to mutation. So that means to say insertion of one or more bases in a DNA molecule is called base addition, or loss of one or more things in it is deletion. So either addition or deletion. So as a result, mutation occurs. If lose or insertion, if a lose or insertion of base exceeds beyond 3, then it leads to frame shift mutation. Of course, reading frame is completely, it is changed as a result. Then generally proteins produced by frame shift mutations are non-functional, hence Such mutations are more deleterious than those produced by the base substitution. Then transposition. Genome of both prokaryotes and eukaryotes um, contain a variety of DNA sequence that can move from one location in the genome to another. Such DNA sequences are known as, of course, just now your friend was asking about this information. So transposable genetic elements, transposons, and their movement, of course, you know, like to and fro in a genome location is called transposition, and integration of a transposable element into a gene often inactivates the gene. So that's the thing. So if this gene encodes an important product, then inactivation of the gene leads to a mutant phenotype. So that's the things. And of course, this transposition, like there they can be considered among the, biological, mutagens. So you have physical, chemical, and biological mutagens. It is not known that many of the classical mutations of the Drosophila maize and the Escherichia coli were result of such transposition. Of course, so later, like people, how it happened. Of course, it was due to this thing, transposition. So that particular things. How it happens? Transposable elements, of course, which can change their things, So they are capable of attaching themselves anywhere in the whole genome. So that is because they carry, because their ability to change their location and get themselves integrated to the genomic DNA, it is through transposase. So the copying and insertion, copy of transposable elements. Here, the gene F interrupted and no longer functional. As a result, what happens? Transposable elements. So they like thing, so they have, their numbers, the places. It is increased as compared to the genes, their position. Suppose if they sit at in between a functional gene and in this case they are showing the two places in this gene. Only a small portion of the gene is left out, which remains non-functional. Then trinucleotide repeat expansion, a sequence of three nucleotides that is repeated too many times in a section of a gene. Example, Fragile X syndrome is the result of an increase in the copy number of CGC trinucleotide repeats in the thing, so FRAXA site, that is the normal 50 copies of copies, but not but mutant 1000 copies of this repeat of X chromosome. Other examples, the of course many in diseases these mutations. For example, some of the examples, you have Huntington disease, Kennedy disease, myotonic dystrophy, then etc. In all these disorders, severity is positively correlated with the copy of number of nucleotides. Repeat expansion mutation in this they are showing. So where initially a trinucleotide is added in the course of replication, it's thing, and in this case, which codes for determine the amino acid. So its numbers, gets increased during replication, repeated replications. Repeated trinucleotide adds a string of glutamines to the proteins. Application of mutations. Some mutations have beneficial effects and are useful in crop improvement. Of course, people, not only crop improvement, of course, breeders, they were using through mutation, like the breeding lines, they used to develop. So they, by exposing the seeds, the seed material to the mutagenic agents, chemical mutagens, and then or physical mutagens, and then of course, uh, like after sowing, any change particular thing is seen. So like many times, they came across some thing, uh, good transformation, good beneficial things, and which was of course they were made use for the breeding purpose. On the fundamental side, mutation in a gene allows the gene to be identified and studied. Of course, by introducing mutation, the advantage is, of course, which is done by molecular biologists. So you can, by introducing mutation in a particular gene, you can know, its functions. So that is possible, by introducing the mutation at a particular thing. So the role of that particular thing can be found out. That is through mutation, it is possible. Then uh, it we have, the fine structure analysis of the gene. Of course, that is possible uh, through mutation, introducing mutation at the things. Then determination of relation between genes and proteins. That is also, of course, otherwise, suppose gene to mRNA, mRNA to protein, it occurs. So how exactly the gene thinks, the information is carried down the things, So that particular thing is, understanding this particular part, gene and the protein. So that is possible by introducing mutation in the gene level, and of course try to study the things. Then it helps in analysis of biochemical pathways, particularly biochemical mutations. So by, of course, by introducing mutations in certain biochemical pathways, so the thing, they are called as biochemical mutations. Of course, it is used for beneficial purpose also, especially in many drugs, pharmaceutical industries have been using in, then many biochemical thing, like biochemicals. So the which like value-based biochemicals. That case, like by introducing mutation, of course, one can look into whether there is enhanced production or decreased production, or the production loses its like what you call control. So it may, like it is no regulation, it will go on producing the similar kind of product. So as the regulatory thing is mutated, so that kind of things are possible. Then induced mutations are used for analyzing the effects of various types of known alterations in DNA on the expression of the concerned characters. Analyzing the effects of these different things, on the alterations in DNA and expression of the concerned characters. So that is thing, it can be understood by introducing mutation. Such an analysis is called reverse analysis. So with this, so we will, we have completed this chapter. Any questions if you have, you can ask. Otherwise, so we will.
Hello, sir.
Yeah, we will. Okay, thank you all.
ಸರ್ ಅಲ್ಲ ಸರ್, ನಮಗೆ ಮೆಸೇಜ್ ಬಂದಿತ್ತು ಸರ್. ಅಂದ್ರೆ
ಬಂದಿದ್ಯಾ?
ಆಫ್ಲೈನ್ ಎಕ್ಸಾಮೇ ಮಾಡ್ತಾರಂತೆ ಸರ್ ನಮ್ಗೆ.
ಓ, ಇಲ್ಲೇ ಮಾಡ್ತಾರಾ?
ಹಾ ಸರ್, ಹಂಗತೆ ಡೇಟ್ ಗೀಟ್ ಏನ್ ಹೇಳಿಲ್ಲ ಸರ್. ಬರೇ ಏನಂತ ಹಾಕ್ತಿದಾರೆ ಅಂದ್ರೆ ಸರ್, ಆನ್ಲೈನ್ ಎಕ್ಸಾಮ್ ಅಂತೂ ಮಾಡಲ್ಲ.
ನಮ್ಮ ಹೆಚ್.ಓ.ಡಿ ಡಾಕ್ಟರ್ ಈರಣ್ಣನ ಕೇಳ್ತೀನಿ.
ಹಾ ಸರ್.
ಕೇಳಿ ಅದು ಹೇಗೆ
ಸರಿ.
ಎಲ್ಲ ನಾವೇ ಫುಲ್ ಮಾಡೋದಂತ, ಫುಲ್
ಹ್ಞೂ ಸರ್,
ಮಾಡ್ತೀವಿ.
ಎಲ್ಲ ಇಂಟ್ರಿಮ್ಸ್, ಪ್ರಾಕ್ಟಿಕಲ್ಸ್, ಎಕ್ಸ್ಟರ್ನಲ್, ಇಂಟರ್ನಲ್ಸ್ ಎಲ್ಲ ಆಫ್ಲೈನ್ ಮಾಡ್ತೀವಿ ಅಂತ ಹೇಳಿದಾರೆ ಸರ್.
ಅದೇ ಅದ್ ಬಂದ್ರೆ ಅದ್ರ ಪ್ರಕಾರ ಮಾಡೋಣ. ಈಗ ನೀವು ಈಗ ಅದು ಕ್ವಶನ್ ಕೊಡ್ತೀವಿ ಏನಿ,
ಹಾ ಸರ್.
ಅದ ಆನ್ಸರ್ ಆದ್ಮೇಲೆ ಅಲ್ಲಿ ಮಾಡ್ಕೊಳ್ತೀರಿ. ಇದ್ರದ್ದು ಪೇಪರ್ಸ್ ಎಲ್ಲ ಇಟ್ಕೊಂಡಿರ್ಬೇಕು, ಮಾಡ್ತೀರಿ.
ಹಾ ಸರ್.
ಮಾಡಾದ್ಮೇಲೆ ಅದನ್ನ ಅಫ್ಕೋರ್ಸ್ ಯು ಕ್ಯಾನ್ ಇದರಲ್ಲೇ ಮೊಬೈಲಲ್ಲೇ ನೀವು ಇದು ತೆಗೆದು ಫೋಟೋ ತೆಗೆದು ಮತ್ತೆ ಅದನ್ನ ಅಪ್ಲೋಡ್ ಮಾಡಬೇಕು.
ಹಾ ಸರಿ.
ಹಾಗೇನಾ ಅಥವಾ ಹೇಗೆ ಅಂತ? ಈಗ ಲಾಸ್ಟ್ ಟೈಮ್ ಎಲ್ಲ ಈ ಇದರಲ್ಲಿ ಬಯೋ ಫರ್ಟಿಲೈಸರ್ ಇದರಲ್ಲಿ ಮಾಡಿದ್ವಿ, ಹ್ಯಾಂಡ್ಸ್ ಆನ್ ಟ್ರೈನಿಂಗು. ಲಾಸ್ಟ್ ಇಯರ್ ಜೆನೆಟಿಕ್ಸ್ ಕಾಣುತ್ತೆ, ಅದ್ರಲ್ಲೂ ಹಾಗೆ ಮಾಡಿದ್ವಿ, ಮೈಕ್ರೋಬಲ್ ಜೆನೆಟಿಕ್ಸ್. ಈಗ ಅದನ್ನ ನೋಡೋಣ, ನಿಮ್ಗೆ ಏನಾದ್ರು ಇನ್ಫಾರ್ಮೇಶನ್ ಬಂದಿದ್ರೆ ಇಮಿಡಿಯೇಟ್ಲಿ ಕನ್ವೆ ಮಾಡಿ.
ಸರ್ ಅದೇ ಇನ್ಫಾರ್ಮೇಶನ್ ಅಂದ್ರೆ ಸರ್, ಅವರು ಆಫ್ಲೈನ್ ಎಕ್ಸಾಮ್ಸ್ ರೆಡಿ ಆಗಿ ಕೊರೋನಾ ಕಡಿಮೆ ಆದ್ಮೇಲೆ ಆಫ್ಲೈನ್ ಎಕ್ಸಾಮೇ ಮಾಡ್ತೀವಿ ಕರೆಸ್ಕೊಂಡು ಅಂತ ಮೆಸೇಜ್
ಮಾಡಿದಾರ.
ಹಾ ಸರ್, ಹಾಕಿದಾರೆ ಸರ್ ಅಂತ.
ಹಂಗಾರೆ ಯುಲ್ ಬಿ brought back to campus and then the examinations will be arranged ಅಲ್ವಾ?
ಹಾ ಸರ್.
ಅದೇ ಈಗ ಅಷ್ಟೊತ್ತಿಗೆ ಬೇಗ ಇನ್ನು ಮುಗಿಸ್ಬಿಡೋಣ.
ಹಾ ಸರಿ.
ಇವಾಗ ಒಂದ್ 2-3 ಚಾಪ್ಟರ್ ಇದೆ. ಒಂದು ಮೈಕ್ರೋಬಲ್ ಇಂಟ್ರಾಕ್ಷನ್ ಇದೆ, ಇಮ್ಯುನಾಲಜಿ ಇದೆ, ಮತ್ತೊಂದು ಮೈಕ್ರೋ ಆರ್ಗಾನಿಸಮ್ಸ್ ನ್ಯಾಚುರಲ್ ಇಕೋಸಿಸ್ಟಮ್ ಇದೆ. ಒಂದು 2 ಪೋರ್ಷನ್ ಮೈಕ್ರೋಬಿಯಲ್ ಇದು, ಅದು ಬಯೋಕೆಮಿಕಲ್ ಆಕ್ಟಿವಿಟೀಸ್ ಒಂದು, and factors affecting environmental factors affecting microorganisms. ಅದೊಂದು ಲಾಸ್ಟ್ ಎಂದು ನಿಮ್ಗೆ ಎಲ್ಲ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ ತೋರಿಸೋದು. ಅದನ್ನೆಲ್ಲ ಮೊದಲೇ ಮುಗಿಸ್ಕೊಂಡ್ಬಿಟ್ರೆ ಇಟ್ ವುಡ್ ಬಿ ವೆರಿ ಈಸಿ ಫಾರ್ ದೆನ್. ಸೊ ಲೈಕ್ ಅಂಡ್ ಎಕ್ಸಾಮ್ ಈಗ ನಿಮ್ದು ಆಕ್ಚುಲಿ ಯಾವಾಗ ನಿಮ್ದು ಸೆಮಿಸ್ಟರ್ ಕ್ಲೋಸ್ ಆಗ್ಬೇಕಿತ್ತು?
ಸರ್ ಜುಲೈ 5 ಸರ್.
ಅಂದ್ರೆ ಇನ್ನೂ ಒಂದು ತಿಂಗಳು ಟೈಮ್ ಇರುತ್ತೆ ಅಲ್ವಾ?
ಇದೆ ಸರ್. ಹಾ ಸರ್.
ಬಂದ ತಕ್ಷಣ ಮಿಡ್ ಟರ್ಮ್ ಮಾಡ್ತೀವಿ.
ಹಾ ಸರ್.
ಅಂದ್ರೆ ಎಕ್ಸ್ಟರ್ನಲ್ ಮಾಡ್ತಾರಾ?
ಹಾ ಸರ್, ಹೌದು ಸರ್. ಎಕ್ಸ್ಟರ್ನಲ್ ಅವರೇ
ನಾವು
ಮಾಡ್ತಾರೆ.
ಪ್ರಾಕ್ಟಿಕಲ್ ಎಕ್ಸಾಮ್ ಆಸ್ ಯೂಶುವಲ್ ಮಾಡಿ ಸಬ್ಮಿಟ್ ಮಾಡೋದು. ಪ್ರಾಕ್ಟಿಕಲ್ ಎಕ್ಸಾಮು ರೆಕಾರ್ಡ್ಸ್ ಎಲ್ಲ.
ಹಾ ಸರ್.
ಅದೇ ರೆಕಾರ್ಡ್ಸ್ ಎಲ್ಲ ಅಫ್ಕೋರ್ಸ್ ಇಲ್ಲಿ ಬಂದ್ಮೇಲೆ ಮಾಡ್ಬಿಡ್ಬೋದು ಒಂದು ವೀಕಲ್ಲಿ.
ಮಾಡ್ಬೇಕು ಸರ್. ಹಾ ಸರ್.
ಇವಾಗೆಲ್ಲ ಪ್ರೋಟೋಕಾಲ್ಸ್ ಕೊಟ್ಟಿರ್ತಾರೆ, ನಿಮ್ ಸೀನಿಯರ್ ಫ್ರೆಂಡ್ಸ್ ಇರುತ್ತಲ್ಲ,
ಹಾ ಸರ್.
ಡಾಕ್ಟರ್ ಸಿಫ್ಟ್ ಎಲ್ಲ ಕಳಿಸ್ತಿದ್ದಾರೆ. ಅದನ್ನೆಲ್ಲ ಮುಗಿಸ್ಕೊಂಡು
ಹಾ
ಅದನ್ನ
ಸರ್.
ಇದ್ ಮಾಡಿ ಅವೆಲ್ಲ ಇದು ಟಿಲ್ ಎಂಡ್ ಅದನ್ನ ಇದ್ ಮಾಡ್ಬಿಡಿ. ಕೊನೆಗೆ
ಹಾ ಸರ್.
ಅದಾದ ಕೂಡ ಒಂದು ನಿಮ್ಗೆ ಮೊದಲೇ ಒಂದಿಷ್ಟು ಸ್ಯಾಂಪಲ್ಸ್ ಎಲ್ಲ ಇದ್ ಮಾಡ್ತೀವಿ. ಅದ ಇದ್ ಮಾಡ್ಬಿಟ್ಟು ನಿಮ್ಗೆ ಅಷ್ಟರಲ್ಲಿ ಅದು ಲಾರ್ಜ್ ಅಂದ್ರೆ ಒಂದು 1 to 2 3 4 ಟೈಮ್ಸ್ ಜಾಸ್ತಿ ನಂಬರ್ಸ್ ಎಲ್ಲ ಇದ್ರ ಸ್ಯಾಂಪಲ್ಸ್ ಎಲ್ಲ ತೋರಿಸ್ತೀವಿ.
ಹಾ
ಅಲ್ಲಿ
ಸರ್.
ಯಾವ್ದು ಬೇಕಾದ್ರೂ ಬರ್ಬೋದು. ಸಿಂಪಲ್ ಆಗಿರುತ್ತೆ ಎಕ್ಸಾಮ್. ಪ್ರಾಕ್ಟಿಕಲ್ ಎಕ್ಸಾಮ್ ಮಾಡಿ
ಹಾ ಸರ್.
ಮಾಡ್ತೀವಿ.
ಸರಿ ಸರ್.
ಬಟ್ ಇಲ್ಲಿ ಕೋವಿಡ್ ಸೆಂಟರ್ ಮಾಡ್ಯಾರಲ್ಲ, ಏನ್ ಮಾಡ್ತಾರೆ ಇವರು ಕಳ್ಳರು?
ಸರ್ ಅದೇ ಸರ್, ಏನ್ ನಮ್ಗೂ ಗೊತ್ತಾಗ್ತಿಲ್ಲ ಸರ್.
ಅದಕ್ಕೆ ಈಗ ಫಸ್ಟ್ ಇನ್ನು ಮುಗಿಸ್ಕೊಂಡ್ಬಿಡೋಣ
ಸರಿ ಸರ್.
ಎಲ್ಲ
ಸರಿ ಸರ್.
ಕ್ಲಾಸ್ಗಳದು. ಅಟ್ಲೀಸ್ಟ್ ಮೊದಲೇ ಅಡ್ವಾನ್ಸ್ ಆಗಿ ನಿಮ್ಗೆ ಫಸ್ಟ್ ಕ್ಲಾಸ್ ಮುಗಿಸಿ ಬಿಗಿನಿಂಗ್ ಇಂದ ಚಾಪ್ಟರ್ ವೈಸ್ ಕ್ವಶನ್ಸ್
ಹಾ ಸರ್.
ಕ್ವಶನ್ಸ್ ಹೇಳ್ತಾ ಹೋಗ್ತೀವಿ. ನೀವು ಅವತ್ತು ನೋಟ್ ಬುಕ್ ತರ್ಕೊಂಡ್ಬಿಟ್ರೆ ಎಲ್ಲ ಬರ್ಕೊಂಡ ಹೋಗಿ.
ಹೌದು
ಆಯ್ತಾ?
ಸರ್.
ಅದು ಮೋಸ್ಟ್ಲಿ 1 hour ಒಂದು ಫುಲ್ ಕ್ಲಾಸ್ ಬೇಕಾಗುತ್ತೆ ಅದಕ್ಕೆ ಟು ಗಿವ್
ಹಾ ಸರ್.
ಕ್ವಶನ್.
ಹಾ
ಸೊ
ಸರ್.
ಅದನ್ನ
ಹಾ ಸರ್.
ಮಾಡೋಣ. ಅದ್ ಮಾಡ್ಕೊಂಡ್ಬಿಟ್ರೆ ಅದನ್ನ ಈಗ ಊರ್ನಲ್ಲೇ ಅದನ್ನು ಆನ್ಸರ್ಗೆ ಎಲ್ಲ ಮಾಡ್ಕೊಬೋದು.
ಹಾ ಸರ್, ಅದನ್ನ ಮಾಡ್ಬೋದು ಸರ್.
ಅದನ್ನೆಲ್ಲ ಮಾಡಿ ರೆಡಿ ಇಟ್ಕೊಂಡ್ಬಿಟ್ರೆ ಇಲ್ಲಿ ಬರ್ತಾನೆ ಮಿಡ್ ಟರ್ಮ್ ಎಕ್ಸಾಮಿಗೆ ಅಫ್ಕೋರ್ಸ್ ಮೊದಲೇ ಇಲ್ಲಿ ಬಂದ್ಮೇಲೆ ಹೇಳಿರ್ತೀವಿ ನಿಮ್ಗೆಲ್ಲ.
ಹಾ ಸರ್.
ಬಟ್ ಇವೆ ಯಾವಾಗ ಆಗುತ್ತೋ ಇಲ್ಲಿ ಅದೇನೆ ಅಲ್ಲಿ ಕೋವಿಡ್ ಸೆಂಟ್ರಿಗೆ ತಗೊಂಡಿದ್ದಾರೆ. ಅದೇ ರೆಸ್ಟ್ ಅದೇನೋ ಸೆಂಟರ್ ಅಂತ.
ಹ್ಮ್ ಸರ್.
ಆ, ಎಲ್ಲಿ ಹುಡುಗರನ್ನ ಕರ್ಸಿ ಪಾಪ ಎಲ್ಲಿ ಇರ್ತಾರಪ್ಪ ಇವ್ರು? ನೋಡೋಣ. ಆಯ್ತಾ?
ಸರಿ ಸರ್. ಸರಿ ಸರ್.
ಓಕೆ ಓಕೆ.
ಥ್ಯಾಂಕ್ ಯು ಸರ್.
ಥ್ಯಾಂಕ್ ಯು. ಬಾಯ್.